Hello, good evening everyone. Welcome you all to the week 10 NPTEL TA session. Today we'll be discussing some questions and assignments on engineered immune cell for cancer therapy. Okay, so let's start it then. Or I'll request you to wait for 2 minutes and let other people join. Then we can start the class. Okay, and please let me know if my screen is not visible or if I am not audible to you people. Okay, just wait for 2 to 3 minutes. Okay, let's start then. So, today we'll be talking about cancer and uh, different microenvironment, tumor microenvironment, and how immune cells can be engineered to cure cancer. Okay, so here in this picture, you are seeing this tumor microenvironment. So, what is this? So, at first, tumor means it is uncontrolled cell growth, right? Everyone knows that if the cell is growing in an uncontrollably manner, then it is called a tumor. And after that, so how this tumor gets affected by the surrounding environment, that is includes the blood vessels, immune cells, and other stromal cells also present. So different types of cells, that is the host cells we are talking about, right? The host cells and the non-tumor cells, how they are affecting the tumor. Okay, and how they are affecting this tumor. So they are continuously interacting with the tumor cells. So this interaction of non-tumor cells with the tumor cells is basically called tumor microenvironment. Fine. So here in this figure, give me a minute. So here in this figure, you can see different types of cells are present. Okay. So it is the representation of tumor cells. Here it is written. Okay. These are the normal cells and these are the tumor cells. And different types of immune cells including dendritic cell, macrophages, CD4 T cell, NK cell, they are inhibiting their each other's expression and leading to the tumor's progression. Okay, and sometimes they inhibit the tumors also, but in case of their inter-signaling pathway, the tumor gets benefited. Okay, so how they are interacting, so and this non-tumor cells interaction is called tumor microenvironment. Basically, we will be talking about it more. It has been established that evading immune destruction is one of the characteristics of cancer. Okay, that's what we are discussing about. And they can inhibit immune effector cells and cause immune tolerance through secretion of extrinsic factors affecting tumor microenvironment. And they include stromal cells, immune cells, and other cells, including tumor cells. Basically, macrophages and T cells are most distorted immune system components. So, why they are included mostly in the tumor microenvironment? So, basically, what we can say, that macrophages are these phagocytes which which engulf the dead cells or tumor cells. Okay, and place it or present is at the MHC2, right? MHC1 class 1 and MHC class 2, you have heard about it. So MHC class 1 is expressed in all types of cells, but MHC class 2 is expressed in antigen presenting cells. Okay, so basically what they do, they present this some antigens like some tumor cells to the T cell, and then T cell get activated and kills the uh, tumor cells. Fine. So MHC class 2, these are, we'll be talking about it more. I'm just explaining you the whole thing in a certain manner. MHC class 2 expression is found on APCs, that is antigen presenting cells, and these antigen presenting cells include macrophages, B cells, and dendritic cells. Fine. So this tumor associated macrophages, they support tumorigenesis. Now you can get it why we are talking about macrophages in so basically the work of macrophage is to engulf the tumor cells and to kill them. But in case of tumor associated macrophages, it supports tumorigenesis. Tumorigenesis means new tumor formation, okay, new tumor generation, and metastasis, and inhibit anti-tumor responses by releasing different types of uh, factors like IL-6, VEGF-8, TGF-beta, and all like that. Okay, so that's why this macrophages and T cells are very distorted in case of tumor microenvironment. And T cells anti-tumor activity and metabolic state is disrupted by the immune modulatory cytokines. Okay, so now you can get it. basically the work of T cells is to kill the tumor cells, but now they are this anti-tumor activity also disrupted by immune modulatory cytokines. So now you can understand this whole figure. The macrophages, and this is the macrophages, and these are the T cells. So this macrophages and T cells are basically getting deactivated or disrupted by their own cell own cell of the microenvironment. Okay, so that's why these two are called most distorted in case of tumor microenvironment. Fine. I guess everyone is clear with tumor microenvironment. This is the basic concept of uh, cancer cell interaction with host non-tumor cells. Let's talk about it more in our upcoming slides. So I want you to answer this question first. So the question is, the process of spreading to other organs is called death and is a major cause of death from cancer. Is it mutation, metastasizing, tumorigenesis, or neoplasm? Please try to answer this question, then we will be discussing it in a short time. I am giving you 2 minutes. You can write the answer in the chat box.
हम्म शाबाश
Please mute yourself.
आंटी टीचर ने साल इतनी ज्यादा पेशे वगैरह भी डलवाएंगे वो भी डालूं कि ना डालूं नहीं वो रहने दो अरे तो मैंने नंबर काट लिए नहीं काटती काट लिए तो कोई बात नहीं फिर क्या हुआ ड्यूरिंग स्कूल लेसन ये इक्वल
Okay, fine. Let's discuss it then. So I got your answers, and it is tumorigenesis, right? So let me explain it more. Then I think you are not understanding the thing. See, tumorigenesis means generation of new tumor in the body. Okay, tumorigenesis, as the name suggests, generation of new tumor. And here is the question: the process of spreading to other organs. So when one tumor is already generated and it is spreading to other organs, okay, so what is it called? First thing, let's eliminate, let's get understand this each and every option. First thing is mutation. So what is mutation? Mutation means any sequential change or any change in the nucleotide sequence of our genetic material leads to production of new or variation of genetic products. Okay, So that's called mutation. Any change in the nucleotide sequence leads to production of different forms of protein, and it regulates different functions also. Fine. Sometimes mutations are beneficial, and but sometimes mutations are harmful too. Okay. Next thing is metastasizing, or you can say it as metastasis. Fine. So metastasis means when one cancer cell is already present in your body and it is spreading to different organs. Okay. For example, if some patient has cancer or tumors in their kidney, okay, but it is spreading to nearby organs like liver and other parts of your body, then this is called metastasis. Okay. Let's see neoplasm. So what is neoplasm? Neoplasm means this is the you can say synonym of cancer. Okay. Generation of uncontrolled cell growth or tumor or cancer is called neoplasm. Fine. Let's talk about it more. So in this figure you can see the progression of cancer cells into the whole body. Okay. And this progression is called metastasizing or metastasis. So according to WHO, cancer or neoplasm or malignant tumor. So what is the meaning of malignant tumor? Same thing, malignant tumor means tumors are of two types. Okay, benign tumor and malignant tumor. Those tumors that, I guess everyone understood the meaning of tumor. Tumor means uncontrolled cell growth. Okay, formation of a tissue like substance, and it leads to uncontrolled and generates in a uncontrollable manner. Fine. So this benign tumor, it resides at one place only. For example, if a patient has tumor in the liver, then it resides in liver only and it doesn't spread to different organs, then it is called benign tumor. Okay. And when it is called malignant, when it starts to spreading different organs, it is called malignant tumor. This tumor itself is called malignant tumor or cancer, but the process of spreading is called metastasis or metastasizing. Okay. Is a large group of diseases that can start in almost any organ or tissue of the body when abnormal cells grow uncontrollably and go beyond their usual boundaries to invade adjoining parts of the body and spread to other organs. So when we are talking about cancer, then we are talking about malignant tumors. Fine. And the process of spreading to other organs is called metastasizing and is major problem of death from cancer. And cancer immunotherapy exploits the body's own immune system to fight against cancer. Okay, so today we are talking about immunotherapy, right? We'll move into it little by little. So in the figure itself, see here, this is the primary tumor formation. Look at this. This red thing is the tumor, and then it leads to local invasion. Local invasion means you can see this fibrillar structures has generated from this whole tumor. Then it is called intra-invasion. Intra means different, it just affecting the local cells only. Intra means it is affecting the blood cells, blood vessel nearby it. Okay. Next, extra survival in the circulation. So you know, uh, blood flow through the blood vessels. So when these cancer cells or malignant tumor cells move into the blood vessel, they survive through the circulation. Okay, moving its speed and its components, they get survived in the circulation. Arrest at a distant organ site, get get arrest at particular organ site, and then lead to extravasion, means leaving the blood vessel and moving out to the organ itself. This leads to micrometastasis formation. Metastasis formation. That means these are the cells, and look at the tumor size. This cell gets extravasated from the blood vessel to the nearby organ, and now it is increasing its shape and getting generating more and more via uncontrollable manner. So see here, this is the small tumor. Next, it leads to metastatic colonization. It leads to a big tumor and then clinically detectable macroscopic metastasis. See here, in a human body, these red dots are shown as tumors or malignant tumors. So now you can understand the process of metastasis. At first, it is a tumor at a particular organ, but now it is spreading whole all over the body, and after that only you can detect it. That's why cancers are deadly, and even there are lot of medications and therapeutics available. People it leads to death at a early age. Okay, so I guess everyone understood the process of metastasis. Let's move to the next question. If you have any question or any doubt regarding this, you can put one question in the chat box. Okay, fine. Let's solve this question. M1 macrophages are able to, so earlier we are talking about macrophages and T cells in the tumor microenvironment. So basically, you can call it as a tumor microenvironment. The associated cells outside the tumor is called tumor microenvironment, including the tumor cells. And this M1 macrophages, so macrophages get maturated via two subtypes, M1 stage and M2 stage. So here we are talking about M1 macrophages, and they can able to release pro-inflammatory cytokines such as IL-12, IFN gamma, IL-1, IL-23 and INOS, or re-educate the DC, dendritic cell or CD4 T cells, deactivate CD8 T cells, and as a result promote an immune response against tumor and prevent tumor progression. So which one it will be? There are three options. So macrophages can do which one? Everyone please try to answer this question. Write your answer in the chat box. Everyone read the question carefully again and answer the question. Okay, let's discuss it then. See here. This is the structure of M1 macrophage. And this is the structure of M2 macrophage. So what is this M1, M2? These stimulants present in TME, that is tumor microenvironment, such as cytokines, chemokines, and growth factors, determine the polarization of macrophages and their differentiation towards M1 and M2 subtypes. Okay, so how they got M1 and M2? This macrophages gets maturated to product to different forms, that is M1 macrophage and different M2 macrophage. So there might be some reason behind this M1 maturation and M2 maturation. So let's talk about it more. See here, I was talking about this MHC class 2 antigen presenting cell. Fine, and its function is pro-inflammatory activity, microbial and tumor activity, tissue damage. So this is the function of M1 macrophage. Got it? Next is M2 macrophage. Its function is anti-inflammatory activity by phagocytosis capacity, tissue regeneration and repair, angiogenesis and immunomodulation, tumor formation and progression. So, which one helps the uh, formation of tumor and progression? This M2 macrophage. Okay, and this angiogenesis. So, what happens in case of cancer or metastatic or malignant tumors? There is one point you need to note, that is, it requires angiogenesis. Angiogenesis means new blood vessel formation. Okay, new blood vessel formation is known as angiogenesis. So basically, this malignant tumor cells spread from one organ to other organ via blood vessels. So that's why, and to spread their tissue, spread their cells to different organs, they require new blood vessels, and also for nutrition. Okay, because this cell is dividing continuously, that means it require lot of nutrition to increase its size. So for that also it requires lot of nutrition and want to spread its cells to all over the body. That's why new blood vessel formation happens, and this process is called angiogenesis. Got it? Anti-inflammatory activity, everyone knows about it. Phagocytosis, phagocytosis means killing and engulfing the dead cells. Okay, so this is the function of M1 macrophage and this is the function of M2 macrophage. So what is this? These are NF kappa B, start 1, start 3, start 6. They are the signaling pathways they are associated with. Got it? And these are the cytokines they produced. This includes produce a lot of cytokines. Here you can see this, and these are the antigen presenting cell. Sorry, MSC class 2 INOS receptors, different types of receptor, T cell receptors and other receptors, and CD cells has been expressed via M1 macrophage and M2 macrophage. Okay, but our question aims to M1 macrophages only. So now we will talk about M1 macrophage. They are able to release pro-inflammatory cytokines such as IL-12, IFN gamma, IL-1, IL-23, INOS, like this. Okay, these are pro-inflammatory cytokines. Re-educate the DC, that is dendritic cells, and CD4 T cells. Fine. Activate CD8 T cells, and as a result promote an immune response against the tumor and prevent tumor progression. Okay, so this M2 macrophage, it helps in tumor formation and progression. But M1 macrophage, it activates the CD8 T cells and as a result promote immune response against tumor and prevent tumor progression. Fine. Now you get to understood the difference between M1 macrophage and M2 macrophage. So this M1 macrophage generally uh, leads to immune response against tumor progression, but M2 macrophage leads to more tumor generation. Fine. And they produce different type of pro-inflammatory cytokines. Okay, so moving to our question, here you can see the third option, it says deactivate the CD8 T cells, but M1 macrophages activates the CD8 T cells and as a result they promote the immune response. So your answer will be 1 and 2. Which one is it? C. Okay, I guess you understood. Everyone understood these things. What are the difference between M1 macrophage and M2 macrophage and how they are connected with tumor progression or tumor generation or anti-tumor activity. Clear? Okay then. Fine. Let's move to our next question then. So here is our next question. Immune checkpoint inhibitors are designed to reinstate anti-tumor immune responses by interrupting co-inhibitory signaling pathways and to promote immune-mediated elimination of malignant cells, immune cells, tissue cells, or cytokine. I suggest you to read this question again and try to answer this question. Write the answer in the chat box. Everyone, please try to answer this question. Okay, let's discuss it then. I got your answer. It is B or A. Fine, then discuss it. So, in the right hand side figure, you can see that this is a T cell. This is the tumor cell, dendritic cell, and macrophages. How they are connected and how they are interacting with via different signaling mechanism, including different pro-inflammatory cytokines and other CD8 or CD types of T cells. Okay. So immune checkpoints are molecules of co-inhibitory signaling pathways that act to maintain immune tolerance. So what is the meaning of co-inhibitory signaling? This is called co-inhibition. Fine. Inhibiting each other that act to maintain immune tolerance which avoid indiscriminate immune attacks. However, they are often utilized by cancer cells to evade immune surveillance. Okay. Immune checkpoint inhibitors are designed to reinstate anti-tumor response by interrupting co-inhibitory signaling pathways and to promote immune-mediated elimination of malignant cells. Got it? So what is this co-inhibitory signaling? So as the name suggests, it is the meaning of co-inhibition, matlab inhibiting each other. Okay, and they act to maintain immune tolerance, which avoid indiscriminate immune attacks. That means sometimes immune cells attack each other. Okay, and that's why the host organism or host body gets affected by this, and this generally leads to production of different types of diseases like rheumatoid arthritis. Okay, they are this imbalance of immune cells that leads to this type of diseases. Fine. And there are, in case of cancer also, cancer cells use these cells. Okay, cancer cells use different types of this co-inhibitory signaling pathways and this cell to invade their immune system of the host. And for that, different immune checkpoint inhibitors are designed, which reinstate anti-tumor response. So at first, cancer cells use these cells to increase its progression or tumor progression, but immune checkpoint inhibitors, what they do, they reinstate the anti-tumor activity. So what this anti-tumor activity, that means it will kill the tumor cells and reduce the generation of tumor cells. That means it will cure the cancer in the body. Okay, and that's why it leads to immune mediated elimination of malignant cells. Malignant cells, we have talked about it in a quite described manner, so I think everyone understood the meaning of malignant cells. Malignant cells are cancer cells, those cells which can invade from one organ to another organ in the human body or different any type of organism. Okay, so your answer is, now you can get it, these are the malignant cells. Fine. So I hope you understood the meaning of immune checkpoint inhibitors and how it is connected to anti-tumor activity. Fine. Let's talk about it more. So cancer vaccine. I don't know if everyone heard about it or not. So let's discuss about it more. I guess it will be a new thing to know because cancer vaccines are not quite old to discuss. It is in the new generation only. So cancer vaccines take advantage of tumor associated antigens or tumor specific antigens to stimulate the immune system. Fine. Especially a robust long-lasting immune response of CD8 T cells to inhibit the death of tumor cells. This question is taken from your lecture slides of week 10. You can get this whole thing from your lecture slides only. Okay. Please read this question again and again and then try to answer this. Everyone, please try to answer this question. I hope you need to read this question carefully, then can everyone can answer. Okay, let's discuss it then. I got answers D, B. Okay, C. Let's see. So, I hope everyone read the question carefully. So, a long-lasting immune response of CD8 T cells to inhibit the death of tumor cells, inhibit the growth of tumor cells. First, let's understand the meaning of each and every term. Okay, inhibit the growth. Yes, we want to inhibit the growth of tumor cells. Next, infection. So tumor cells no not infect, they just invade. And recurrence. Recurrence, what is the meaning of recurrence? Recurrence means generally disease recurrence happens or cancer recurrence happens. So at first recurrence, the meaning is the disease comes again and again after giving medication. So if a patient goes through this cancer therapeutics or different types of medication to or chemotherapy to kill the cancer cells or tumor cells, but after 6 months or 8 months of time period, it is found that the cells again started regenerating and they are producing the tumor, and the cancer again happens again seen in that patient. Okay, so this is called recurrence of disease. Fine, means this disease after being cured for some time, after a certain time period, it comes again in the patient. So this is called recurrence. Next thing is killing. See, inhibit the killing of tumor cells. So do you want to inhibit the killing of tumor cells? If we inhibit the killing means what? Death of tumor cells. If you don't want to kill the tumor cells, then the tumor cells will generate more and more, and that will lead to production of very deadly kind of cancer. Yes or no? Okay, I think you understood. This inhibit the killing. That means if you don't want to kill the cancer cells, then it will be producing malignant tumor, malignant cancer, or death of the patient. Fine. So we want the killing of tumor cells. We don't want tumor cells to grow. Fine. So this term just discard this option B. Understood? Those who answered option B, I guess you are getting my point, that we want to inhibit means what? Decrease or prevent the growth of tumor cells, not killing of tumor cells. Next thing is metastasis. Metastasis, I guess everyone understood. Metastasis means spreading of cancer cells from one organ to another organ. Fine. Recurrence I discussed. Growth, metastasis, immunosuppression. So we inhibit the immunosuppression. Do you want to inhibit the immunosuppression? Immunosuppression means suppressing the immune system. Okay, and we don't want that. We want to activate the immune system so that it can kill the tumor cells. Next, growth, metastasis, and recurrence. So yes, we want to inhibit the growth of tumor cells. We want to inhibit the metastasis and recurrence of tumor cells. So this is your answer. Got it? Let's talk about it more. So cancer vaccine takes advantage of tumor associated antigens or tumor specific antigens. Tumor associated antigens, I guess you understood, that is the tumor cells only, nothing else. These are represented as antigens so that antibodies can be formed against this. To stimulate the immune system, especially a robust or long lasting immune response of CD8 T cells. So why we need cancer vaccine? To long last the immune response and to inhibit the growth, metastasis, and recurrence of tumor cells. According to clinical use of cancer vaccines, they are divided into two categories: preventive and therapeutic. This preventive cancer vaccine aims to prevent tumor occurrence by inducing immune response, while the therapeutic cancer vaccines are designed to eradicate tumor cells by inducing or enhancing the tumor specific immunoreactions. Okay, if you read this sentence again, I guess you understood these things. I don't want to repeat the things, same thing again and again. Okay, so if you have any doubt regarding this, you can ask me in the chat box. Okay, I'll repeat the thing. Fine, everyone understood what we are talking about, how cancer vaccines are helping us to cure the tumor. Next thing, so here is our next question. The efficacy of cancer vaccine primarily depends on immunogenicity, host immunosuppression, preferential expression of tumor antigen, or all. Everyone please try to answer this question. I guess we have discussed more about cancer vaccines to answer this question. So I got answers D and A. Let's talk about it. So the efficacy of cancer vaccine primarily depends on all. Okay, so your option answer is all. That is immunogenicity, host immunosuppression, preferential expression of tumor antigen, okay, and all. That's why the efficacy of cancer vaccine depends upon all of the things, not a single thing. Fine. I guess everyone understood the terms because we have discussed it. Host immunosuppression means how much it is suppressed, or preferential expression of tumor antigen. That means tumor antigen, which tumor antigen is getting expressed, or how much, or if the whole cell is getting act as an antigen, or any part of the cell is acting as an antigen. That's also depends upon the efficacy of cancer vaccine. If we include the preferential expression, that is the tumor antigen is uh, expressing whole in a whole structure, then it will help the cancer vaccine to act in a more efficient way. Let's discuss about it more. So see here, in this figure you can see how these cancer vaccines have been made. Okay, so here, for example, this is the patient and it has tumor. Okay, so remove the tumor cells from the tumor itself and culture it by a cell culture. So these cancer cells from a patient are killed and in some cases broken down into parts. So these tumor cells are broken down into specific parts to increase its expression, or sometimes it is expressed at the whole cell. Fine. And they are cultured. Now these dead cancer cells or components of the dead cancer cells are then combined with adjuvant to make a vaccine. These dead cancer cells are components of dead cancer cells. These are the dotted spots, and this whole thing is called as adjuvant. So what is an adjuvant? Can anyone tell me? Write this in your chat box. What is the thing adjuvant? Okay, fine then. So adjuvants are basically used to increase the expression of antigen. Okay, so vaccines were, so what is the meaning of vaccines? Or vaccines are specifically designed in a way where these dead antigens are presented to the immune cells of host body. Then they get activated, they produce antibodies, and these antibodies production is remained as a memory inside the host body. So when a real antigen or real pathogen comes to the host body or host immune system, then they can easily produce the antibodies against that pathogen. Okay. Yes, a substance or component that is added to the vaccine or medication to enhance the body's immune response. Yes, it's true. So we want to increase the antigen expression inside the human body or host body. That's why these adjuvants are added, so that it can increase the expression of the antigen and the antibodies can easily be produced inside the host body. Fine. And this vaccine stimulate the immune system to respond to antigens. I guess everyone is clear with vaccine. I don't need to repeat it. Still, if you have any doubt, then you can ask. Okay. So these are the lymph nodes which gets activated by this vaccine, and they produce. Why we are talking about lymph nodes? Because lymph nodes are the sites where B cells generally proliferate. Okay. And they lead to production of immune response, and then this immune response or promoted immune response will kill the tumor cells and reduce the tumor activity. Understood? So this is the whole thing called cancer vaccine. So cancer vaccines are basically made from the tumor antigens, and these tumor cells are dead tumor cells, not active tumor cells. Okay, sometimes vaccines are made with partial living. Okay, that means some partial dead organisms or partially dead pathogens can be used for production of vaccine or generation of vaccine. But in case of cancer vaccine, we use dead tumor cells. Fine. And sometimes it is broken into parts to get more expression of certain parts only. Fine. And it, in a brief manner, or we can summarize it as cancer vaccines promotes the immune response, and then this immune cells, that is the T cells, kill the tumor cells. This is the whole thing, and we add the adjuvant to increase the expression. Fine. Okay, then I guess everyone understood. Let's move to our next question then. So this is our next question. ACT method uses which immune cell? ACT means adaptive cell therapy. It uses the B cell, T cell, dendritic cell, or cytokines. Which one is it? Everyone, please try to answer this question, then we will be discussing it in 2 minutes. Okay, let's discuss it then. I got the answers as T cells. Yes, and it's correct. So here is your answer. ACT method, that is adaptive cell therapy. This method uses immune cell, which immune cell? It is T cell. Okay. So generally it is called T cell therapy. So why we are talking about this cancer vaccine or T cell therapy? Because today's session is about using immunotherapy against to cure the cancer. Okay, so that's why we are talking about different types of immune cells which can be used as a therapeutics against cancer, and it can be engineered using genetic engineering. Fine. So earlier we were talking about cancer vaccines. Now we are talking about T cell therapy. So what is it? T cell therapy? Basically, so for example, uh, it is a patient here. So first thing, what we need to do? Tumor or blood samples can be taken. So why blood samples can help? See, in cancer, that is the malignant tumors, it is expressed inside the blood vessels also. Okay, that's why it is dotted all over the body. Actually, these are the T cells, but I am just trying to tell you that in case of malignant tumor or in case of a cancer patient, the tumor cells can be found in the blood vessels also or in the blood samples also. Okay, so we can extract the tumor cells or tumor or blood samples. Fine. So from blood sample and tumor cells, T cells has been isolated, okay, and then T cells multiplied and often their functions are enhanced. So why we are talking about T cells more? Because T cells has the efficiency to kill the tumor cells. So we want to, there are different types of drugs and medications are available, but we want to do it in a natural manner. So this is the natural mechanism present inside our body, right? So except using different types of drug and therapeutics, which are number of uh, extra risk or side effects. Everyone knows the side effects of chemotherapy and others. Okay, so these other therapeutics, including chemotherapy and all, they have large number of side effects. But using T cell therapy, it reduces the side effects and it increases the efficiency because we are using nothing but the T cells of a host body only. Okay, so these T cells are multiplied and often their functions are enhanced, and then they are injected back into the human or human patient as enhanced T cells, and these T cells now can move to different parts of the body, and they can multiply or recognize and kill the cancer cells. Okay, so this whole thing is called T cell therapy. Fine. So basically, to summarize the whole T cell therapy, we can say that we are extracting T cells from the patient only, and in the lab method or different types of enhancing method, we are increasing the efficiency of T cells, and then we inject the T cells again to the patient. These enhanced T cells, they have the efficiency to kill the tumor cells in a more efficient way as comparison to other T cells present inside the patient body. Okay, I guess this summarizes the whole thing. If you have any question, you can ask me. Fine, then let's move to our next question. Yes, but see, it is better to take it from the tumor cells only, na? Tumor cells or blood cells. So where we want the tumor cells, see, in a bone marrow, for example, if your bone marrow is active, that means your bone marrow is mm, not affected by the tumor, not affected by the cancer, then we don't need to activate those T cells. We want to enhance the T cells or T cell function which is deactivated by the tumor. Okay, basically in the tumor microenvironment, there are different T cells and other cells which get immunosuppressed. This cancer suppress their activation or suppress their function. That's why we want to activate the T cells more and more so that they can function it in a better way. Fine. For cancer, we generally take samples from blood or tumor. Yes, bone marrow cell T cells can be used, but it is better to use the tumor cells T cell. Fine. Okay. Thank you. ACT is followed by through the major methods, including, now we have talked about the T cell therapy, that is adaptive cell therapy. So this method is followed through major methods, including using tumor infiltrating lymphocytes, T cell receptors, modified T cell, or engineering chimeric antigen receptor, that is CAR T cells. Which one is it? Or all? Try to answer this question, everyone. Okay, so I got answers. It is D. Okay, D. Yes, it's correct. On. Okay, so ACT, that is adaptive cell therapy, is followed through different measure methods, including tumor infiltrating lymphocytes. We are just talking about it, right, in the previous case. So we are taking tumor or blood samples. Tumor infiltrating lymphocytes, that means those lymphocytes which can infiltrate the tumor. Fine. Next, T cell receptor modified T cells. Yes, we are just trying to enhance the T cells immune function, right? So if you want to enhance the T cell function or enhance the T cells immune function, then obviously you want to modify it. So yes, it's correct. T cell receptors modify T cells. Next is engineered chimeric antigen receptors T cells. This is another method of engineering T cells against cancer therapy, and it is quite widely used right now, and it is successful. So CAR T cells is very important to understand the cancer therapy, and we will discuss about it more in our upcoming slides. Okay, fine. So this is our next question. The heavy chain variable region and light chain variable region, that is the VL, of antibodies are linked by a small segment of polypeptide, CAR T cells, tumor cells, or CD3 delta chain. If you remember the structure of antibody, then you can answer this question. I guess everyone have read about the structure of antibody. Please try to answer this question. If it is a guess, then it will be okay. We will be discussing the whole structure of antibody. Okay then. I guess everyone answered it because it is quite clear via eliminating the options. Fine. So everyone is correct. That is the polypeptide. So let's talk about the structure of antibody. Okay. So this is the whole structure of one antibody, and this whole thing is the antibody binding region, and this thing is called biological activity mediation. Okay, so this is used for binding purpose, and this is for antibody interaction. So it has two chains. One is heavy chain. Okay, another one is light chain. Fine. So I am not discussing each and every part because it will take time for this, and we have a number of questions to discuss. So just basically it has these two chains, heavy chain and light chain, and they are connected via interchain disulfide bonds and intrachain disulfide bonds. This is the hinge region, and these are the structures with hyper variable regions where your splicing mechanism, you have heard about it, right? This splicing mechanism of your mRNA leads to different types of hyper variable changes in this region. Okay, and it is written in quite detailed manner, so you can read about it. I will upload the slide after the class. Okay, fine then. Let's move to our next question. The toxic levels of systematic cytokine release and severe immune cell cross activation in some patients result in the following toxicity. So read this question again and try to answer this question. Take your time, 2 minutes or 3 minutes, but read this question twice or thrice, then I hope you can answer. Okay, so I got only one answer, that is C. I think everyone is confused. Okay, fine. Let's discuss it then. Then we'll know which one is the answer. So, toxic levels of systemic cytokine release and severe immune cell cross activation. So, what is this meaning of these two terms? So, generally, in case of cancer patient, now we are talking about immune cell therapy, right? So, in patients, we doctors use this method of immunotherapy to cure their cancer or kill the tumor cells, but in some cases they leads to different types of toxicities or side effects, that is systemic immune cytokine release. So generally in case of cancer, I have talked about it again in our previous slides, that this macrophages, M1 macrophage and M2 macrophage and others, they release several pro-inflammatory cytokines, and these cytokines reduce the activity of other T cells and other immune cells, and leads to the progression of tumor, that leads to malignant tumor or cancer progression. Fine. So in this case, sometimes we use systemic release of cytokines. That is, cytokine is released in a certain manner into the body, or severe immune cell cross activation. Earlier we are talking about this T cell activation, right? In case of T cell therapy or adaptive cell therapy, we are talking about cell cross activation. That means we are enhancing the activation or enhancing the function of T cells in some patients. Fine. So these two things are mentioned here. So in case of that, this patient sometimes uh, go through some toxicities. So what are these toxicities? Let's talk about it. See here. This toxic level of systemic cytokine release and severe immune cell cross activation in patient result in following toxicities. Which one is it? Cytokine release syndrome, that is associated with supraphysiologic cytokine production and massive in vivo T cell expansion. Okay, so it is the name suggests, I think everyone is clear. Sometimes it happens supraphysiologic cytokine production, that means cytokine is produced in a large number, and that leads to massive in vivo T cell expansion. T cell is expanded more from its regular size. That is also harmful for the human body. Okay, next, hemophagocytic lymphohistiocytosis, that means macrophage activation syndrome. Okay, this is also called macrophage activation syndrome. That is a severe hyperinflammatory syndrome characterized by CRS, that is cytokine release syndrome, and combination of elevated serum ferritin and hemophagocytes, renal failure. Do you understand the meaning of hemophagocytosis? Hemo means what? Hemoglobins. That means this phagocytosis happens in our red blood cells. Fine. That's why it's called hemophagocytosis. Renal failure, liver enzymes, splenomegaly. Splenomegaly means your spleen increases its in size. Why? Can anyone tell me, do you know about this? Fine, fine. Let's talk about it. So, this T cell maturation and B cell maturation happens inside the body at first, right? So, spleen is one maturation site. Yes. So, if we increase the number of T cells, then the spleen's activation or spleen function will more and more, and that will lead to this splenomegaly. Okay. Another is pulmonary edema. Edema means swelling. Pulmonary means our respiratory tract or leading to the lungs. Connected with the lungs, it's called pulmonary tract. Pulmonary edema or absence of NK cell activity. NK cell means natural killer cells. They are specifically present inside our immune system to kill the uh, outer cells, outside cells. Fine. So I guess everyone understood the terms one by one. Okay. So hemophagocytic means the killing of the red blood cells or the blood cells present, phagocytosis of blood cells. Lymphohistiocytosis, that is, it is connected with your lymph nodes. Hist means your tissue. So as the name suggests, it what happens, the hyperactivation happens, which leads to failure of that organ. Okay. Next, immune effector cell associated neurotoxicity syndrome. So that is, the name suggests neurotoxicity, that is it associated with your nervous system, and elevated cerebrospinal fluid cytokine levels and blood brain barrier disruption. So what happens when we when there is this systemic cytokine release or increase of cytokine levels, because we are providing from outside, right? So when it happens like this, and it this whole mechanism gets some kind of abruption or disruption, this leads to your increased cytokine level in your cerebrospinal fluid, which leads to blood brain barrier disruption. So this is termed as ICANS. So now you know, if your immune system is not working properly, or if it is slight bit or a little bit dysregulated, then it can lead to different types of diseases including cancers. Okay, so everyone is clear with all these terms. So your answer will be D, all of the above. Fine. Okay, then let's move to our next question. So we are talking about immunocell therapy to cure cancer. Okay. Next is an application of CRISPR in which effect of T cell function on cancer immunotherapy in presence or absence of a gene is investigated, and the results may use for development of further immunotherapeutic purpose. So so this is the mechanism of a certain thing or certain screening method. So what is the name of that screening method? Loss of function screening, knocking out granulocyte macrophage colony stimulating factor GM-CSF, or TME, or CAR screening, CAR. Read this question twice and then try to answer this. Okay, then let's discuss about it. So an application of CRISPR. So if we are talking about CRISPR, so what is it? That means we are talking about genetic engineering or gene editing, right? So what is this effect of gene editing? where in which effect of T cell function on cancer immunotherapy in presence or absence of a gene. We want to know the effect of T cell therapy in presence or absence of a gene, which is investigated for further use. So what is this called? It is called loss of function screening. So if you know about CRISPR, as we have already discussed in our previous TA sessions, so if you have not attended that session, you can go through the YouTube videos where you will find out the CRISPR-Cas TA session. Then if you want to go through it, then you will know that this CRISPR method is used for gene editing or specifically for gene knockout. And in that case, if you want to know the function of a specific gene knockout is specifically preferred. If we knock out that particular gene, then we will know if this is responsible for any important function or it is lethal or whatever. Okay, we will know about that gene. So this is called loss of function screening. Let's talk about more. So basically, loss of function screening is another application of CRISPR in which effect of T cell function on cancer immunotherapy in presence or absence of a gene is investigated and the results used for development of further immunotherapeutic approaches. Okay, and to elucidate immunomodulatory impacts of combinationally role of anti-cancer or cytotoxic properties of effector T cells, and to explore mechanism of CAR T cell cytotoxicity, a detailed in vitro loss of function screening by means of CRISPR was conducted, which is explained in your lecture slides. Okay, so basically you can see here the whole thing is written in kind of a schematic representation. This is uh, this is connected with T cell, and this left part showing the gene editing thing and right part shows the different types of delivery technologies. So how gene editing can happen? Using ZFN zinc finger nucleases. Okay, so this zinc finger nucleases the you cut your DNA and makes a double stranded break, and this double stranded break can be mm, joined together via your NHEJ, that is non-homologous end joining method, or homologous recombination method. Fine. So this one method is zinc finger nuclease method. Another is talent method. Here also this nuclease cuts the DNA at two sides, making a double stranded break, followed by the NHEJ and HR method. And CRISPR-Cas is also used for this. Okay, there are different types of gene editing methods we have discussed. From that, ZFN, TALEN, and CRISPR-Cas is specifically used for tumorigenesis, mutagenesis or specific mutagenesis. And this is the method of CRISPR-Cas9, where this sgRNA, that is the guide RNA, is specifically used with Cas9 nuclease. Okay, and when this sgRNA, that is the guide RNA, forms a double-stranded structure, then the Cas9 will cut the DNA at that particular site. Okay, fine then. I guess you understood the gene editing method. Let's talk about the delivery technology. Delivery can be using by the viral vectors. Okay, viral vectors like adenovirus vectors can be used to deliver this gene editing, this engineered genes, electroporation, cell squeezing, or nanoparticle method. In nanoparticle method, lipids, gold, or polymer can be used. Specifically, nanoparticle particle method is now enhancing in the research field. Okay, and there is this electroporation method is also there, and different kinds of adenoviral vectors are also present. Fine. So this is the whole thing of T cell engineering. That is, we are talking about genetic engineering. This class whole thing is about genetic engineering. So here, using different types of ZFN, TELN, and CRISPR-Cas9 technology, we can engineer the T cells, and then we perform the specific function we want. If you want the function of T cell in presence or absence of a gene or whatever it is related to gene editing, then that also can be done. Okay, fine then. This is your next question. The first clinical trial testing the safety and feasibility of CRISPR-Cas9 editing T cell receptor T cell in patient with refractory cancer was reported in which year? Try to answer this question. Okay, so this is your answer, that is 2020. Okay, so here is another question regarding T cell engineering. Candidate target antigens that are used for T cell receptor engineering T cell treatment require three features if they are to be utilized. Read the question carefully and try to answer it. I guess you can take 2 minutes for this question. Okay, let's discuss it. So candidate target antigens that are used for T cell engineering, and they must be have some features, that is they must be selectively expressed in tumors, not in normal tissues, right? Yes, of course. We want the T cell engineered T cell treatment to be present or to be specified for tumor cells, not normal tissues. Okay. Next, they are related to oncogenesis, tumor addition. Okay. Next, they are able to evoke a T cell response, that is immunogenicity. So these three things is important for your T cell engineering to be highly effective and to be successful. Fine. So your option will be all. Okay. Next, let's discuss about it more, the T cell engineering cells. So for example, here it is the patient here, and we extracted blood from that patient. Okay, this is the tumor, it is shown in pink color, isolated from the blood samples. These are the T cells, that is natural T cell receptor, and scientists genetically engineer the T cells to have T cell receptors that recognize tumors. Now you can understand why we are doing this T cell engineering. Okay, so specific some T cells are present which cannot recognize the T cells. That's why their expression or their activation or function is specifically low. Okay, so that's why scientists genetically engineer the T cells to tumor targeting. Okay, they specifically target the tumors, and this T cell engineered T cells are multiplied using cell culture system. Next, it is delivered back into the patient as a living drug. In case of cancer vaccine, remember it, we were using the dead tumor cells. Okay, not living. We are using the dead tumor cells. But in case of T cell engineering T cells, we are using the living engineered tumors, sorry, T cells to deliver into the patient, that is act as a living drug, and this engineered T cells continue to multiply within the patient where they recognize and kill the cancer cells. So this is a simplified version of schematic representation showing T cell engineered T cell. There are lot of function and molecular mechanisms are present behind this, but we are trying to understand it in a basic manner. To understand the basic concept is more important rather than going through the molecular thing. You can go to the molecular thing after this. Okay, so I hope you understand the thing why we are doing this T cell engineering and cancer vaccine. So today we talked about different types of immunotherapies, including cancer vaccines, T cell engineered T cells, and our effective macrophages, and different types of engineering methods to provide sufficient immune response to our patient body to act to activate it anti-tumor activity. Fine. So this is all for today. Thank you for joining the session. If you have any doubt, you can ask me here. Okay. If you don't have any doubt, then we can end this session here. Thank you for joining again. We'll meet in next Monday. Okay then. Thank you.